everyone, I'm Dr. Saif and I'm back again with another video. I would like to start this video by asking you, look around you, your parents, your grandparents, even that wise old tree in your village square. What do they all have in common? They're aging. We often see aging as a slow, visible decline. A few more grey hairs here and there, a slower walk, and maybe a few aches and pains here and there. But what if I told you that beneath all this, complex, high-stakes biochemical drama is playing out in every single cell of your body? It's like a grand old Bollywood movie, but at a molecular level. We are talking about the biochemistry of aging in this video. So how would you define aging? It's actually a very simple definition. Aging can be defined as a progressive decline in the physiological function. This leads to an increased susceptibility to disease and ultimately a journey towards the inevitable. It's not a disease itself, but a major risk factor for almost every chronic disease we encounter in our wards, from diabetes to heart disease to neurodegeneration. We also need to understand why does it matter? Why does the study of aging matter? Because understanding the molecular basis will allow us to develop strategies to promote healthy aging, ensuring our elders live not just longer but better. We start our journey with the genetic factors. And when we talk about the genetic factors, the number one genetic factor associated with aging is telomeres. Imagine our DNA is like a sacred book, and this book is like the instruction manual of all the cells. With age, what happens is this book gets damaged and misread. Here we encounter something called telomere shortening. Now, what are telomeres exactly? Think of them as the ends of your chromosomes, like the plastic tips on shoelaces, or even better, the boundary ropes on a cricket pitch. These are telomeres. These are repetitive DNA sequences. And by repetitive, I mean specifically TTAGGG sequences in human beings. They protect the actual genetic information during replication. But there's a problem. Our DNA polymerase is a bit like a lazy contractor. It can't copy the very ends of linear chromosomes perfectly. So with each cell division, these telomeres get shorter. Once they hit a critical length, the cell shouts, "Bas ho gaya," and enters replicative senescence. This is actually called the Hayflick Limit. And this is the point that the cells shout, "Bas ho gaya," and they enter replicative senescence. And it stops dividing. However, there is an exception here. There is an enzyme called telomerase. It is a reverse transcriptase, and it acts like a ground staff, adding back those boundary ropes, allowing for indefinite divisions. So telomerase basically adds back these telomeres, allowing for indefinite division. That's why cancer cells are immortal. Suddenly come to cancer cells here. When we were talking about the normal pathway, this is a deviation from what we were talking. In cancer, this telomerase is quite active, and it keeps bringing the telomeres back. And that is why the cancer cells never stop dividing. They keep dividing, they never die. Now, in rare genetic disorders like Progeria, so this Progeria is Hutchinson-Gilford Progeria syndrome. It's a syndrome in which children age rapidly, often due to defects affecting the telomere maintenance or DNA repair. They show premature graying, skin atrophy, and cardiovascular disease. Here's a cool picture which shows how the telomeres shorten. And as they shorten, the cell division also gets reduced. This is the telomere. And it shortens over time as the cells keep dividing. The next concept we come to is DNA damage and impaired repair. Our DNA is constantly bombarded from metabolic byproducts like reactive oxygen species to UV radiation, environmental toxins, and even errors during normal replication. Cells have an incredible arsenal of DNA repair mechanisms. These include base excision repair, or nucleotide excision repair, or double-stranded break repair. All of these repairs are essential. For example, base excision repair fixes small damages. Enzymes like DNA glycosylases, AP endonucleases, and DNA polymerases and ligases take part here. Then there is nucleotide excision repair, which repairs bulky damage. Think UV-induced pyrimidine dimers. Various enzymes work here like XPC, XPA, XPF, and DNA polymerase and ligase as well. Then there is double-stranded break repair. This is the most critical, like repairing a broken bridge. And it is of two main types, non-homologous end joining and homologous recombination. So homologous recombination involves the BRCA 1 and 2 genes, which is crucial for breast cancer risk. Now, the aging connection here is, with age, our cellular repair crew gets tired and of course less efficient. All this DNA damage keeps accumulating, leading to genomic instability. This genomic instability is also considered to be one of the risk factors for cancer development. That is why aging is even associated with the development of cancer. Now let's move on to the next concept. This is the concept of epigenetic alterations. What is this epigenetics? Epigenetics is about changes in gene expression without altering the actual DNA sequence. So we see a chromosome here, and then chromatin, and then the gene and the DNA. Epigenetic mutations cause very small alterations without altering the actual DNA sequence. So the DNA sequence remains the same. Only certain alterations occur. It's like having the same holy book, which is the DNA, but different priests. So they have different interpretations and they highlight certain verses and ignore others. Now the key mechanisms involved in epigenetic mutations include DNA methylation. Then there is histone modification. Now in histone modifications, there are HATs and HDACs. This is a bit advanced, but let's just see each one of them. DNA methylation, just like the name suggests, it's like adding a methyl group to cytosine bases. What this usually does is it silences the genes, like putting a do not disturb sign on a gene. Now if we recall, histones are the proteins, these are histones, around which our DNA is wrapped. There are certain chemical tags like acetylation, methylation, or phosphorylation. These tags on these histones, which are proteins, act like volume control knobs or light switches. So these tags have an important role. They act as modulators of the genes. This brings us to HATs, which is histone acetyl transferases. These enzymes add acetyl group, generally opening up the chromatin, and this promotes gene expression. Then there are HDACs, which are histone deacetylases and sirtuins. They remove acetyl groups, generally compacting the chromatin. And whenever the chromatin gets compact, this represses gene expression. So you can see both of them have completely opposite effects. Now you might be thinking, we are talking about aging, so how does this all? We see an epigenetic drift, which is a fancy word of saying there is global loss of methylation, but at the same time there is specific hypermethylation elsewhere, and this leads to altered histone patterns. So all these histone patterns will get altered. This leads to inappropriate gene activation or silencing, which messes up the cell function. It's like the conductor of a classical Indian orchestra losing the score, and the instruments are all there, but the harmony is lost. This is what is observed in aging. The next concept we come to is mitochondrial dysfunction. Now, if you recall, the mitochondria are the powerhouse of the cell. They generate ATP through something called oxidative phosphorylation, which is all orchestrated by the electron transport chain. Now, what happens is, just like an old engine of a car, even the mitochondria produce fumes. Now, these fumes are nothing but reactive oxygen species, or ROS. This occurs due to electron leakage. Now, the vicious circle develops where these reactive oxygen species, they damage the mitochondrial DNA, proteins, and lipids. And this further impairs the electron transport chain, which in turn leads to even more reactive oxygen species production. And all this will eventually lead to decrease in ATP. This theory is basically called the free radical theory of aging. Now, one thing to note here is, so cells normally have a system of destruction or clearing of damaged mitochondria, which is called mitophagy. What happens with age is this cleanup mechanism also slows down, and this leads to accumulation of dysfunctional mitochondria. Now, scientifically, this can be associated with fatigue, muscle weakness, or energy deficit in elderly population. As age progresses, all these things start to creep in. The next is cellular senescence. Now, what are senescent cells? These cells are not dead, but they have hit a cellular retirement age. They have stopped dividing, which means they have reached cell cycle arrest, but are metabolically active and resist apoptosis. Now, if you remember, apoptosis is programmed cell death. They just hang around like that one Uncle G who refuses to leave the family gathering. SASP is their defining feature, or it is basically short form for senescence associated secretory phenotype. These Uncle G cells don't just sit, they gossip, they create a cocktail of pro-inflammatory cytokines. If you remember, the pro-inflammatory cytokines are interleukin 6, interleukin 1, etc. Other than this, they also secrete chemokines, growth factors, and proteases. This is the SASP. Even though it sounds pretty bad, senescence actually evolved as a good thing. It evolved as a tumor suppressor mechanism, which helped in stopping damaged cells from becoming cancerous. But their accumulation and the SASP in aged tissues contributes to chronic low-grade inflammation. Now, this chronic low-grade inflammation is what is called inflammaging. It's like a small constant fire burning in your tissues, contributing to almost every age-related disease. Think of chronic joint pain, atherosclerosis, or even some cancers in the elderly. Senescent cells and their SASPs are often silently fueling this fire. Next, there is proteostasis or protein aggregates. Our cells are constantly making, folding, modifying, and degrading proteins. This dynamic balance is called proteostasis, which ensures proteins are functional. Think of it as a busy factory, which is constantly producing, quality checking, and discarding bad batches. Now, for all these processes, there are certain key players. These include chaperones, ubiquitin proteasome system, or in short UPS, and autophagy. Chaperone proteins include HSP 70 or 90, and they are the quality control inspectors. They help proteins fold correctly. The UPS or ubiquitin proteasome system tags misfolded or damaged proteins with ubiquitin, which is a molecular rejection sticker. It sends them to the 26S proteasome for degradation, which basically shreds them up. And then autophagy is a lysosome dependent process, which is also involved in degradation. Now, how is all of this connected to aging? What happens with age is the efficiency of chaperones, UPS, and autophagy declines. This leads to accumulation of misfolded proteins along with aggregated proteins, and both of these are extremely toxic. Now, the clinical pearl here is neurodegenerative diseases. Can you recall of any neurodegenerative disease? Yeah, Alzheimer's disease, which is basically accumulation of amyloid beta or tau proteins. And Parkinson's is accumulation of alpha-synuclein aggregates. So you can see here how proteostasis is so important, as it plays a role in the pathophysiology of these very important neurodegenerative disorders. Now, the last thing we talk about in this video is the various theories of aging. Now, the first theory is the theory that there are various pathways that sense nutrient availability, and they dictate whether the cell should grow or repair. Now, these pathways include the mTOR pathway or the AMPK pathway. The mTOR pathway basically stands for mechanistic target of rapamycin. It is acting like a growth promoter. It's like a central regulator of cell growth, proliferation, and metabolism, and it is activated by abundant nutrients like amino acids, glucose, and growth factors. It promotes anabolic processes like protein synthesis and lipid synthesis, and it inhibits catabolic processes like autophagy. Chronic activation or overactivation of mTOR is actually linked to accelerated aging, and there's a theory that actually inhibiting mTOR can extend the lifespan. Next, there is the AMPK pathway, which is AMP activated protein kinase. It is the energy saver. It is activated by low energy states. Low energy states are basically high AMP is to ATP ratio. There is less ATP, so that means less energy. When it detects this state, it promotes catabolism, which is glucose uptake or fatty acid oxidation, and that in turn increases ATP, and it inhibits anabolic processes. Other than this, it is known to activate autophagy and mitochondrial biogenesis. AMPK activation is considered to be anti-aging, which is basically opposite to that of mTOR. Now, a small caveat here is, have you heard of the drug metformin? So metformin is known to activate AMPK, very mildly of course. So it is playing a role in anti-aging. Now, let's go to the next theory, which is that of inflammaging. A sterile, chronic, low-grade systemic inflammation is called inflammaging, and it increases with age. It's not an acute infection, but a persistent low-level irritation in your body, like a lingering cough after the monsoon season. Accumulation of SASPs and senescent cells or DAMPs, which is damage-associated molecular patterns, along with gut microbiome dysbiosis and activation of nuclear factor kappa B pathway, which is like a master switch of inflammaging. It increases inflammation. The clinical significance here is that inflammaging contributes to almost all age-related diseases, which includes cardiovascular diseases, neurodegenerative, metabolic syndrome, arthritis, and even increased susceptibility to infections in the elderly. Now, the final theory I'm talking about in this video is the glycation and advanced glycation end products theory. AGEs, or advanced glycation end products, are formed through a non-enzymatic reaction. So they are produced through a non-enzymatic reaction between reducing sugars, proteins, lipids, or nucleic acids. Think of it like slow cooking sugar and milk to make sticky caramel. It's the Maillard reaction. Now, the consequences of these advanced glycation end products is that they cross-link proteins, making tissues stiff. Now, if tissues become very stiff, they become dysfunctional. Think of rigid blood vessels, stiff collagen, etc. They also bind to receptors. These receptors are called RAGEs, or receptors for advanced glycation end products. These are present on cell surfaces and they trigger inflammation and stress. Now, AGEs play a major role in diabetes mellitus, where high glucose levels accelerate AGE formation, leading to complications like diabetic nephropathy, which is kidney damage, retinopathy, eye damage, and neuropathy, which is nerve damage. It's literally like sweet poison causing sticky damage. Now that we have come to the end of this video, I would like to give you a high yield summary, like always. So aging is a multifactorial process which is driven by the accumulation of molecular and cellular damage. So we talked about telomere shortening, DNA damage, and epigenetic alterations, which compromise the cell's blueprint and its interpretation. Then we saw cellular changes. These were mitochondrial dysfunction and cellular senescence, along with impaired proteostasis. And here we also learned about inflammaging, which is chronic inflammation, and accumulation of toxic protein aggregates, which can lead to various neurodegenerative disorders. Then we talked about a couple of theories, and in this we talked about pathways. We saw two major pathways, which were the mTOR and the AMPK pathway. Then we saw the theory of inflammaging and advanced glycation end products. Now, understanding all these molecular mechanisms can help develop therapies to promote healthy aging and combat age-related diseases. The fight against aging isn't about finding a fountain of youth. It's about meticulously understanding each molecular player and pathway and learning how to gently move them back into harmony. Thank you for staying till the end and watching this video. Hope you enjoyed it and hope you learned a lot. And I hope to see you in the next one.